What I'm going to talk to you about today, I mean, our program is really broad, so with Dr. Rutberg's help, we narrowed the focus a little bit, so I hope this is appropriate for this audience and something that you'll find interesting as part of what we do at the AZA Reproductive Management Center. Infertility has become a big deal in zoos. And it wasn't recognized until just a few years ago, and we played a part in that. But the problem of failing reproduction, just reproductive failures without knowing why, was first published by Lees and Wilkins in 2009 for zoo communities around the world, so the global zoo associations. So it wasn't just a US problem, and so we started looking more specifically at what might be happening. So I work with the Reproductive Management Center. The Population Management Center is also American Zoo and Aquarium Association Center. There are two of them. So population management has to do with genetics. They're based at the Lincoln Park Zoo in Chicago, and then the reproductive physiology and contraceptions at the St. Louis Zoo. So they did an analysis a few years ago and found that of their breeding recommendations, only about 25% were resulting in births. And so that is startling, but my next sort of parenthetic statement there, taking into account, once a pair has bred, an individual has basically left its genes in the population, it will then be less likely to get another breeding recommendation. It has contributed to the population. The ones that didn't reproduce are the ones that keep getting repeated recommendations. So they're the ones that are of compromised fertility to begin with. So over time in a population, that number gets worse and worse. So what we're dealing with now is that we have a lot of captive populations in zoos that have reached that mature stage. The population has reached that mature stage where most of the breeding recommendations are going to individuals that are of compromised fertility. But that also means these programs aren't meeting their goals, and that has to do with gene diversity for the population, so genetic health of the population, demography, just that you have a somewhat appropriate proportion of males and females and age structure, and then sustainable population numbers. And that's the concern that hit us hardest as reproductive physiologists, this lack of sustainability. And taken further, this could mean that there are species that could go extinct in zoos. Now, this is not extinct overall, but it's backwards of where zoos have been historically in taking pride in and taking very seriously an effort to have the captive population be a backup resource for animals in the wild that are facing very real threats that we all know about from other sources. But that we could have some of these species not being able to make it in zoos because of reproductive problems was startling to us. And just in the last few years as well, something else happened, and IUCN, the International Union for Conservation of Nature, based in Switzerland, it's the biggest conservation organization in the world, partnered with the Global Zoo Association, the World Association of Zoos and Aquariums, WAZA, and has worked out a very elaborate set of, it's like a flow diagram, where in a species recovery would captive breeding become important? How would that happen? How would it be identified? How would that be managed? And as the captive population then being considered part of the global population in the wild as well as in captivity, and looking at those as complementary with animals going in and out of captivity as needed. So that puts even more pressure on zoos to step up and figure out what's going wrong with reproductive processes in some of these species. So the Reproductive Management Center mission, I have to put this up here, manage reproduction for animal well-being and conservation as a service to AZA institutions. And this is just a quick overview of what the center is like, then I want to move on, especially because the program coordinator used to work here, Monica McDonald. So those of you who are younger probably don't know her, but some of you who've been around here for a while may have known her in the past. But probably more importantly is that we have a very large advisory board, about 30 people from different disciplines that provide information that we need, given the range of species differences that we have to deal with, and the different kinds of problems that do come up. Two of the positions, the program coordinator and program analyst, have salaries paid by AZA. The advisory board is voluntary, as many of them are. And then the director and endocrinologist are employed by the zoo. We have partners, the Reproductive Health Surveillance Program at Michigan State University, the vet school, the pathologist Dalen Agnew runs that. Annika Moresco, who's now at Denver Zoo, works with him as well. And we have a partner, like a sister organization in Europe that is started to do advising for contraception in Europe, and that's important because they have different products available than we have here in the US, so they tailor recommendations for the market from Europe. And we also have now a joint web, an MOU and a web-based data entry system. And we recently, in the last year or so, started a new collaborative initiative within AZA. This is a photo from our strategic planning meeting in St. Louis. And what's surprising, AZA has all these committees and scientific advisory groups, and these are the names of those groups that are working with us, and specialists who tackle those particular kinds of problems. But there was no venue or mechanism for them to work together to exchange information. So that's what we're providing for them. In fact, we were on a conference call yesterday for an hour with updates from each of these groups, just so each knows what the other is doing and they can work together on more things where that would be beneficial. And so the RMC role in general in these programs is to help those programs reach their genetic goals, but we have to look at this in a sort of two-pronged approach. So we have to enhance reproduction in some individuals, but we have to limit reproduction in others. And so for enhancing fertility, I have at the top of the list because that's at the top of our agenda right now, but also providing proper social groups. And I put mate choice here because that's one of my special interests. I really think we need to be doing more in zoos to offer mate choice. And then still using assisted reproduction, but I always want to mention this when I'm talking at universities because especially, you're a vet school, not animal science, but especially in animal science programs, students get excited, as do faculty, about the very latest, newest kind of cloning or whatever it might be. This is what's going to save endangered species. That's not going to happen in my lifetime. I mean, maybe we're looking 50 years from now that many of those more advanced techniques could actually be put into practice. Right now, all we need is, that's an oversimplification. In most cases, what we need right now is just plain old artificial insemination. That will solve most of our problems in terms of assisted reproduction. But we also need more systematic gene banking. And of course, that saves genes for the future, but it also can facilitate moving, especially moving sperm, so you don't have to do animal transfers to make pairings. So we coordinate then with the Population Management Center in Chicago, and this is ongoing, but for each species, about once a year, this process is put into play. And so they use information from studbooks, which is basically just pedigrees, and they have computer software, the data go into that, and breeding recommendations come out the other end. There are people who understand how that happens. It's computer dating for animals in zoos. And the breeding recommendations are almost exclusively based on the genetics of the current population. So how diverse is the current population, without taking into account all of the factors that might influence reproductive success. So starting about 2 years ago, we began, in fact, some of our staff are up at the Lincoln Park Zoo today working through some of those things. We need to be partnering with them because their recommendations are for naught if reproduction isn't successful. So we recognize from both sides we need to do this better. So in any given year then, there are animals that receive a breeding recommendation. But for that species, the ones not getting a breeding recommendation are basically being given a recommendation not to breed. So that's where contraception can come in. And we provide information on contraception for certain, but also more and more now, since we morphed from being the Contraception Center to being the Reproductive Management Center, working more on enhancing reproduction as well. So Dr. Rutberg gave you this information before, so I move on, but why use contraception in zoos? And that may be obvious to you, but the main thing for us is being able to maintain animals in natural social groupings. So as pairs or family groups or larger social groups, while still achieving these breeding recommendations, so that the individual animals you want to breed are the ones that are fertile in that group, and the ones that don't have a breeding recommendation then are not. And so we manage population genetics, so equalizing founder representation, that means every population that exists in zoos is there because of a certain number of individual founders that came out of the wild to provide that genetic foundation for them. And that can be anywhere from 10 or 12 up to 50 or so. Most of those occasions were in the early 1900s, and so we're working with what we were given, that 100 years ago or so when those animals were brought in. But we also want to reduce the production of surplus animals, and surplus in this case means genetically surplus to the population. They're not needed for the genetic health of the population. They're not needed to be a breeder. But we also worry about surplus in terms of, and that we really should come up with a better term, that zoos have a finite amount of space, finite resources, and even if we had more space, we would still reach capacity. We can only properly take care of a certain number of individuals at each institution, so we have to limit the numbers. And the importance of natural social groups should be obvious, but it's a better message for people visiting zoos. In some cases, that's the only exposure people are going to have to that species, and so wanting to see them in as natural way as possible is really important. And as I mentioned before, it's better for the animals that don't receive a breeding recommendation not to be pulled off and isolated somewhere for the breeding season or for the year, but to be allowed to stay in that group. So we have a web page, and it's open, not password protected. If you want to go look at what's there, you're welcome to. The address is there, stlzoo.org/contraception. And so there are recommendations for all the taxonomic groups that we have to treat, and complete product descriptions, how to apply them, where to get them, how to use them. And this information is being updated all the time as new information comes in to us. And this is what the landing page looks like. As I said, you're welcome to check it out. And the methods that we're using right now in zoos, for the most part, synthetic progestins. I'll go over each of these in a little bit more detail. The estrogen progestin pill, that's what's produced for humans. GnRH agonists, the PZP vaccine. And all of these, except the GnRH agonists, are female-directed, and that really represents what's true for humans as well. Those are the products that we have available. So we have MGA, melangestrol acetate. These are all progestins. available as implants, as a liquid that can be added to food, or even already incorporated into an herbivore diet for hoof stock. Medroxyprogesterone acetate, that's Depo-Provera. We do use that, it's injectable. Magestrol acetate, Ovaban is now off the market in the US. I keep hoping it's going to come back, but as far as I know it's not. We get it as a generic now. And then Altrenagest, which is Regimate. So the MGA as implants were first used in zoo animals, in lions and tigers in 1975, so this goes way back. And have now been used in virtually every mammalian taxa, at least the ones that exist in zoos. And they're very effective. Depo-Provera used mostly in animals that it would be difficult to get an implant into, to have a suture line with an implant inserted. Hippos, marine mammals. Lemurs, because they're seasonal, so if you put an implant in, it's good for 2 years. You don't need 2 years' worth in a seasonal breeder. And then postpartum injection for little callitrichids works really well. While baby's clinging to mom, they have a postpartum estrus within about a week of giving birth, so that gets them through that time. The Ovaban pills were originally used in bears, and that we noticed that when we first started our program. And I'm not even sure how that got started. Somebody decided to do it and others picked it up. That's not the case now. Now we use it mostly to suppress the flare with GnRH agonists. I'll get back to that when I talk about them. Regimate, just like in horses, wild equids seem not to respond to any progesterone and Altrenagest, so that would be used in zebras and asses, and then in marine mammals because it's an oral solution, and so for marine mammals that are trained to come up and take a fish or something, you can dose them like that. So, progestin contraceptives, if you've got the right dose and you actually get it into them, they're virtually 100% effective, and they've got a high reversal rate, so we like that a lot. The implants, though, that's a celastic implant that you see up there, requires surgical insertion, maybe just an inch, but still, you've got a suture line, you maybe have some sutures, animals pick at it, it can be a problem. The feed and liquid require daily administration, or you can have a surprise estrus, an ovulation, and that can be a problem, especially giraffes. We have that problem. They're very picky eaters. Depo-Provera, and this is true for humans as well, the period of efficacy is from about 2 months up to 2 years. So if you're trying to formulate breeding plans, that can be a real problem. But we're more worried about side effects. So weight gain, true for humans as well. If you're on a progestin-only contraceptive, you're likely to gain some weight. It can block parturition in many species. Primates seem to be the exception, but in others it's the case. You can't treat a pregnant female. Then endometrial hyperplasia has been identified in some primate species, and hydrometra in ungulates, but that appears in both cases to regress when treatment ends. So we don't worry about it a lot. But for carnivores, there is a serious, potentially at least, serious problem. The uterus and mammary tissue of carnivores reacts differently to progestin products. And so mammary and uterine tumors are most commonly reported, and a lot of this literature is domestic dogs and cats. So we draw on that, but we've also got quite a bit of data for wild felids and wild canids in addition. But more recently, we've also become concerned about endometrial hyperplasia that contributes to infertility. So it's not at what might be considered a disease state yet, but it can impair implantation or pregnancy maintenance. And of course, it can favor the production or the growth of bacteria and lead to pyometra. So there are painted dogs, also called African wild dogs, Cape hunting dogs, are really prone to this problem. So this has become a big problem for us. And at higher doses or long-term treatment, even diabetes, we've seen that mostly in canids. So we need to find alternatives, especially for carnivores. Birth control pills, great for apes. In fact, they're used commonly in apes and zoos, but they're still steroid hormones. Still not a good option for carnivores. The PZP vaccine doesn't work well in most carnivores. Bears may be an exception. There's some growing data for bears that maybe that's going to be an alternative, but for the most part, effective in ungulates. The GnRH agonists, and in particular in this case, I'm talking about suprelorin, which is a product out of Australia. The generic is deslorelin. It's not commercially available in the US, so we import it under an arrangement with Verbach and FDA, and we essentially run a multi-center research trial. So our center is able to get permission to do that, and we work with more than 100 zoos. An advantage is that it can be effective in all mammals. Even though it's a protein molecule, it's similar across mammals. It's also similar in males and females. It's GnRH is the master hormone that controls reproductive hormones in both males and females. And it's tiny. It's the size of a grain of rice, so it can be inserted with a trocar, no surgical incision. It's not, there we go. A potential problem is that as an agonist, it first stimulates the system. And so there's what's sometimes called a flare. And this can cause estrus and ovulation that can be fertile in females. In fact, there's a version called Ovuplant that's not on the market in the US right now. We import it to use in wolves. That's how we synchronize estrus and ovulation in wolves in the US, by getting Ovuplant, which is a short release, like a bolus release form of deslorelin. But if it's given chronically, then in a sustained release implant, it will cause downregulation at the pituitary, so you don't then get LH, and you still get some FSH production that's independent of GnRH stimulation, but it downregulates enough that downstream you're not getting production of estrogen and progesterone in females, and you're not getting, I think that's my next slide, and you're not getting testosterone in males, so you can interrupt reproduction. There are two formulations, one a minimum of 6-month efficacy and the other a minimum of 12-month efficacy. Yes, aren't they the cutest things? I know it's really awful to consider having wildlife as pets. If I was going to break that rule, this is what it would be. Meerkats are the cutest things. Okay, back to being serious. But the problem with suprelorin, that the minimums are accurate, but we're seeing some individuals, especially the big cats like lions, suppressed 5 years or even a little bit more with one dosing. And so I can't say just one implant because we actually calculate dose by multiples of implants. So a larger animal might get 2 or 3 implants at one time rather than just one. They only make one size implant. So that, again, as with Depo-Provera, that unpredictable time to reversal is a real problem in the genetic management. So, well, what about just separating males from females? I mean, that's what people were saying to us. Okay, especially the old animal science folks, just separate them. And honestly, in some of the ungulates where males and females aren't naturally together in the wild, that's maybe not such a bad option. You just take the male out of the herd during the breeding season. That can work. And maybe that's safer. You're not adding hormones or altering hormones. But okay, I like to think of this, I don't need job security anymore, but it's job security for the people who are still there. Species differences always come around to bite us. So this happened, I guess, somewhere around like 2009, 2010. I realized we were getting more and more reports, just spontaneous reports to us, especially from people holding African painted dogs and red wolves, which is a different species from the gray wolf, of pyometra. And it's not that they were reporting a contraceptive problem, but they didn't know who else to call. We were still the contraception center there, but we were reproductive physiologists, so they called us. You know, what might be going on here? But it was coming in sporadically, and then one day I thought, there's a trend, there's something going on, I'm really worried about this. We should at least look at the history of these individuals collectively and see if there might be a contraceptive involved, because that's our responsibility. If contraceptives are causing problems, we need to get on that. So we ran a big study of these 7 canid managed programs in AZA zoos. And so these are the players. So here is Mexican wolf, fennec fox, also really cute. And in fact, they are in the pet market. There are private breeders of fennec foxes in the US that, and we actually work with them because we admit fully, they're better at breeding fennec foxes than we are. That's their there are 2 of them. That's their specialization, that's what they focused on, and they really know what they're doing. So even though there's controversy about wildlife pets, if we look at this honestly and holistically, that maybe there's something going on there we need to pay attention to. Okay, back to, here's swift fox, bush dogs, maned wolf, here's that painted dog, and red wolf. So 2 very dedicated, hardworking graduate students compiled reproductive histories, contraceptive use histories, and compared that to reams of pathology reports that were sent to us for every animal that had been, every female in those programs since 1980. So it took 2 students to get through all of that. And they ran then a multivariate analysis looking at factors that might be related to endometrial hyperplasia or with pyometra. And that's been published in Zoo Biology. And so the model results then for risk factors showed that separating males from females, so that was the reproductive history part. We could look at how many years between litters, just looking at stud books. So if females were left barren for a certain number of years, was there a problem? It turns out it's equivalent to progestin contraceptive treatment for canids. And the reason that may be the case, and since you're vet students, maybe you've had enough repro, especially of dogs, that across all canids, if a female ovulates, she goes through what we call an obligate pseudopregnancy. Now, I understand that veterinarians usually just use the term pseudopregnancy if there are clinical signs, but endocrinologically, hormonally, it is a pseudopregnancy. It's indistinguishable between pregnant and non-pregnant females, except by the hormone prolactin, not prolactin, relaxin is higher in pregnant females. But in terms of progesterone and estrogen, the uterus of a non-pregnant female is being exposed for 2 months to elevated progesterone for certain, estrogen early on. So estrogen priming followed by 2 months of progesterone. And in fact, if you look at pyometra data, most likely time for a female to be diagnosed is in or at the end of that period of time, that 2 months. Okay, so separating male from female was no better than progestin contraceptives. And there were species differences in that risk, really profound species differences. Painted dogs, it was up around 25% of the population that we had records for were experiencing pyometra. And red wolves was in the teens, and fennec and swift foxes were something like 2 or 3%, really low. And we don't think this is a difference in the way data were collected. It's done the same way. I mean, the way stud book data are collected and the way contraceptive reports are sent to us. So we think this is truly a species difference. And we continue to get reports on, and we're working more and more with painted dog folks. In fact, last week, a female at the Kansas City Zoo had to be spayed, a painted dog. She had pyometra. And so we get one of those calls every few months on a painted dog. What we did notice, but we didn't have enough cases to do statistical analysis, is that suprelorin, which is that GnRH agonist, if you give it with, and this may seem counterintuitive, but Ovaban is a progestin, but if you give that orally a week before and a week after the implant goes in, you prevent the stimulation phase. So you have 2 weeks of a little bit of progestin, but you don't have 2 months of elevated, really elevated progesterone. So you basically get immediate downregulation of the system. So right now, no female that was given that regimen has shown up, of all the canids, has shown up with pyometra. We can't say that's never going to happen, but it's suggestive, but there haven't been enough cases for statistical analysis yet. What about other species? Is this just canids? So we did a lit review and brought some colleagues in to help work on that. And so cheetahs, 2 publications were already out there. And they didn't look at it the same way we did. They looked at like how many years a female was just left barren and what was the likelihood she would give birth, or even what did her uterus or ovaries look like, depending on the study. And it was similar to what we were finding with canids. Rhinos and elephants, excuse me, really different species, excuse me, from the carnivores even. Endometrial hyperplasia, but more commonly leiomyomas, the fibroids, were occurring in females that weren't bred until they got to be older. So if they were like a 25-year-old rhino or elephant that had not reproduced, was likely to have fibroids. Even a colony it was an all-female colony of bats, and they had an all-female colony because they didn't know how to control reproduction. So they just didn't put any males in. And then they realized that a lot of those females were turning up with uterine pathology on necropsy. And so they published that. And we continue to find reports and some of it hasn't been done as properly analyzed, like statistically analyzed reports, but a lot of anecdotal reports that are accruing enough now for ungulates that we're becoming concerned more about them. But that is in terms of not reproducing for a long time. But this seems to not be as, I don't want to say there's no problem in primates. That would make me nervous to say there's no problem. But we can't find much, if any, problem with primates. So something different is going on. And whenever any hyperplasia is noticed, then it tends to regress when the treatment is stopped. So we really are back to, carnivores are the big problem, and let's keep an eye on ungulates. But what we can see is that females need to establish fertility early by breeding early on, and to be able to reproduce regularly so they don't go through these long periods of time when the uterus the lining may overgrow. Progestin contraceptives are attractive again, though, because they reverse so easily. And I recently did a really detailed review of the literature for dogs and cats. And there is a growing movement. There's a veterinarian in Italy, Romagnoli, who has a really interesting review paper from a couple of years ago. And he very carefully looked at doses in studies going back to the 1960s with cats. And there is definitely a dose effect. And so at a lower dose, it appears that these progestins can be safer. There hasn't been a comparable study for dogs, but European vets have been using Depo-Provera in dogs for decades, and they start during deep anestrus and give a low dose, and they're not seeing these side effects. So we're looking again at judiciously using progestins. And we know suprelorin is safer, but we do have that reversibility problem. So now we have created something called lifetime reproductive planning. And we, as the title of my talk, use it or lose it. That's the sort of cute way to look at it. But to incorporate the idea of letting a female establish fertility by breeding her not long after puberty, an alternative might be breed early and often. So this is some possible scenarios, and for carnivores, I've written litters, that you could just use something like Suprelorin early on if she's not perhaps very genetically valuable, but you want to hold her in reserve just in case you might ever need to breed her. Or if she's important, that maybe you let her breed, like do her complete genetic contribution to the population, right out of the gate. Let her have 3 litters in a row and then either spay her or put her on Suprelorin. Or if you want to space births, really good data from felids, wild felids like lions and tigers and jags, is that uterine pathology doesn't become a significant problem until and this was on using MGA implants, until it's 4 years. Now, at that point, it can be really serious uterine pathology, but at 2 years, things look pretty good. Now, there could be individual differences, and you have to be careful, but we're thinking one possible scenario could be use it for 2 or fewer years, but allowing litters in between basically cleans the uterus again and sets it back at 0, so our pathologists tell us. So, yes, that's, from a reproductive point of view, that could solve the problem. But we had to talk to the population geneticists, who are concerned, of course, about what this could do to their world, but then we remind them, but in your world right now, you've got animals not reproducing at all, and you're losing their genes entirely. So we reach a compromise. And so we're working with them then. In fact, 2 of them that do computer programming for that computer dating system, and working through various scenarios to see, like those 3 that I showed. If you use that in the population for all females for X number of years, what would your population look like? If you assumed they were successful and they had a normal size litter, what would the population look like? How many times would you need that female to reproduce? What does that do to demography? What does that do to the gene diversity? So that's in process. But something we realized as we were populating the models, and we first did this with fennecs, so that's why their picture is here, is that you need to amass basic reproductive data for the species. And most of this comes out of stud books. And so things like, for fennecs, for instance, what age they are will make a difference in whether they're reproductively successful. Rearing method, there are fennecs that are hand-raised, that pet market. It turns out they reproduce better. The hand-raised ones reproduce better. Crazy. And whether they're sexually experienced. I mean, in subsequent years, if they are, they're more likely to become successful. What's the social grouping like? What location? Like what's the environment? Inside, outside, latitude, climate, that kind of thing. Okay, so that's in process. So stay tuned. And then just to quickly go through tackling infertility and this endometrial hyperplasia. And I'm saying endometrial hyperplasia, not cystic endometrial hyperplasia, because once it's cystic, you can see it with ultrasound. And once it's cystic, we're not sure we can get it to go back. But if we catch it early, there's some hope that we can get it to regress enough to restore fertility. So Bruce Christensen, working with 2 pathologists, did validate a transcervical biopsy technique. He was at Iowa State at that time. He's at UC Davis now, and using dogs that were going to be spayed, and so did the biopsy, then in the next couple of days spayed them and did side-by-side comparisons of biopsy samples and full thickness samples and found no difference. So once the technique was validated, then he started using that in some canids and felids. Here you see a red wolf and a lion on the table. And he's been training other veterinarians around the country that do, if they do transcervical inseminations, they've got the equipment and they're ready to be able to do a transcervical biopsy. So we're getting strategic locations around the country, so if zoos in the area need a biopsy done, they can call on one of these vets. What we're not sure yet is if we can treat endometrial hyperplasia, we think that using something like Suprelorin, if you've prevented that flare, the stimulation phase, just downregulate the uterus for a while and let it recover. We're hoping that will happen. And our priority, as I said before, is with painted dogs because they are the crisis species right now. So how does this relate to captive breeding programs? Well, the reasons for failure that have so far been identified by the Population Management Center: compliance. If pairs aren't been put together at all, that's about 50% of the cases. They're not put together at all. Long list of reasons for that. But reproductive failure, they're put together, but offspring were not produced. So incompatibility or infertility might be the problem, and I'm going to have to hurry a little here, but this is just to point out, zoos have set themselves up inadvertently, unknowingly, for this to happen, because the typical population, founding phase, here are the founders, you breed them up quickly to be sure you've captured all those genes, you get those genes established in the population, and then you get up to carrying capacity and you need to turn them off, and then you end up with infertile animals. So that's what we're working around. So lifetime reproductive planning, we hope will be able to address this, at least will lessen the problem that we're seeing now. And I've decided, I think I'm going to use this slide at the end of every talk I give because this is science. Every time you do a study, you come up with more questions. You answer one and you come up with more. But we've just got so many species that we have to work through, and they react so differently that it will be a challenge, and that could be your future. Thank you.
We have a couple minutes for questions.
Yeah.
Hi, I was hoping you could speak a little bit on mate choice and how that can help increase reproductive ability in animals.
Yeah, well, there are times when pairs are put together and they don't mate. And so we need to figure out why that's happening. And mate choice is certainly one of the possibilities. It again varies by species. So we've been studying cheetahs in particular because we know that female cheetahs are choosy, and trying to figure out what the parameters are, and are there scenarios that will either help us identify, like we're actually using urine as a proxy. Can she sniff urine samples and tell us who she likes, and then we ship those males to her? So you're trying to be inventive, and then working with people to come up with just strategies for presenting choice. Like, how do you do that with, so they're separated, but they have access, and can the males actually display, and can they do what they need to do to impress a female? And then can you tell which one she's actually selecting? So in practice, it's really challenging. So a lot of people in zoos would love to do it. They believe it makes a difference, but they need some more models, which is what we're working on, of how to actually provide that choice and interpret preference.
Has it been proven at all that artificial insemination is less viable than mate choice, or live breeding, I guess you should say?
Live breeding is more, you're more likely to get a pregnancy, absolutely. And especially if you've got frozen semen, then there's even more of a difference. If you've got fresh chilled, you can come close to a natural mating. But it depends on the species, because in some species it's not been worked on at all. So I work with Mexican wolves. We do a lot of that thing, and so I understand those parameters. But yeah, we've got lots of other species that we need to be working with. Yeah.
In your expert opinion, do you see increasing reproductive rates in endangered species a beneficial conservation tool?
I'm sorry, I didn't hear the first thing you said.
So increasing reproductive success of endangered animals in zoos, do you think that's a viable conservation efforts?
So is increasing reproductive success of females in zoos going to contribute to conservation? And yes, and I think it depends on the species to what extent it will help. And I work with Mexican wolves, and they're a great example, because the Mexican wolf program is a complete marriage of captive breeding and animals in the field. So the ones that are out in the field now, which are in Arizona, New Mexico, and then we have partners in Mexico who've reintroduced there, all those animals came from captive breeding. They were extinct in the wild. So the only ones that exist were bred in captivity. But because they predate on cattle, if a wolf is causing problems, they have to capture them and bring them back in, and they come back into the zoo community, and they live in zoos again. And so every year we may be bringing animals in and putting more animals out. So having a genetically viable, healthy population in captivity is critical for Mexican wolves. For some species, it's less important.
Okay, I think we have a class in here at 1, so let's raise the one more time.
Okay.